let's say you work in a clinic and you need to see if someone has an HSV virus, for instance. What we turn to in practice commonly is a technique referred to as ELISA, which is the enzyme-linked immunosorbent assay. And it is a very quick and effective means by which we can screen blood samples, for example, and detect the intensity of some kind of antibody. And so what we're going to do in this video is go over how that process works. And so the first thing we would do if we were in lab is we would take a 96 well plate and coat it with if we were studying the HSV virus, we would place the HSV antigen on the matrix in our 96 well plate. And so after we do that, the next thing we would do is incubate or load in the blood samples. So add blood sample or whatever solution you have you want to screen for into the 96 well plate. And at this point, what would happen is if someone's blood had high levels of the HSV antibody, those antibodies would bind very well to this antigen that you already have in your plate. And so these are referred to as primary antibodies. And so primary antibodies will bind, if they're present, to the antigen that is present in your plate. And so after this step, the next thing we would do is wash away the antibodies without affinity for our target. And this is because blood has antibodies for other antigens not specific to the HSV virus, and then maybe some patients don't actually have the HSV virus, so this step would remove those antibodies. And then after that, what we do is we're going to add in an antibody enzyme complex. And after we add the antibody enzyme complex, it will have a second antibody bind to the primary antibody that's bound to your antigen. And so these complexes will stick to the primary antibodies, and after they do that, this enzyme is a very important component, because this enzyme, when we add a substrate, will act as a biomarker for us. And commonly what will happen is these enzymes will take the substrate and then they'll turn yellow, for instance. And so the final step we get after we visualize is a 96 well. If I just drew a few wells to keep things simple, they might look like this. So we would have some cells that were completely yellow. We'd have some cells that were not yellow at all. Then we'd have some cells with a faint indication of yellow. And so what we would arrive at here is that we could run additional tests or have some kind of good indication as to whether someone might have HSV. And another important thing to note here is whether or not it's yellow, and also the intensity of the yellow color. So if it's a very intense yellow color, it means that there are a lot of primary antibodies present in those kinds of solutions or the blood samples. And so this concludes how we can use ELISA as a good high throughput screening method, as well as a method by which we can quantify the presence of a concentration of some kind of analyte in our solutions. And so I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.